Welcome back to the Deep Dive. Today we are tackling what is perhaps the most fundamental and enduring puzzle in all of science.
The big one.
The big one. How does the physical, the electrochemical hardware of the brain create the richness and complexity of human cognition? The mind itself. We're taking a deep dive into the core architecture of cognitive neuroscience, really distilling the most profound insights.
It really is the ultimate question, isn't it? And the answer keeps shifting. For decades, the field was sort of chasing this holy grail of simple one-to-one maps, find a function, find the spot in the brain. But our sources show the contemporary view is just so much more intricate. The brain isn't a rigid machine. It's both this huge collection of specialized processors and an integrated dynamic network that's constantly reorganizing itself.
The dual identity right there, specialization and integration. That's really the key tension, isn't it? So our mission today is to give you a shortcut to understanding this whole landscape. We're going to unpack the three core dimensions driving the science.
Where are we starting?
We'll start with the huge theoretical debate around modularity. Then we'll get into the method scientists use to map the mind to the brain. And finally, look at the amazing tech that lets us see this all in real time. Let's get started.
Sounds good.
All right, so our first big idea is modularity theory. This really defined how we thought about the mind's structure for a whole generation. It was laid out most formally by the philosopher Jerry Fodor back in 1983.
Right.
He proposed that the mind has these specialized units, or modules, each one dedicated to a specific type of information, like, say, recognizing faces or processing the syntax of a sentence.
And the defining feature of these Fodorian modules wasn't just that they were specialized. It was a much more technical and I think interesting concept, informational encapsulation.
Informational encapsulation. Okay, that sounds complicated.
It does, but it's not so bad.
Let's break it down. Think of it this way. These modules are like locked boxes. They work automatically, they're super fast, but they're sealed off. They operate totally independently of your broader knowledge or your beliefs.
Exactly. Your high-level knowledge about the world, like an optical illusion is false, the lines are the same length, that doesn't stop your visual system, the module, from seeing the illusion.
It just keeps processing it the same way.
It's fast, it's automatic, and it's, kind of stupidly dedicated to its one narrow job. But, and this is crucial, Fodor argued this strict modularity only applied to input systems, so perception and language. He thought central processes like reasoning were just too flexible and holistic to be modular.
But then that idea got picked up and, it got expanded massively by evolutionary psychologists like Lita Cosmides and John Tooby. They proposed something called massive modularity.
And this was a really radical extension. They basically argued that the entire mind, even complex things like social reasoning, is fundamentally modular. Instead of a few input modules, they saw the mind as this huge collection of specialized tools.
Like a Swiss Army knife.
A Swiss Army knife is the classic analogy, yeah. All these tools were sculpted by evolution to solve very specific problems our ancestors faced. So you don't have a general intelligence module, you have a dedicated cheater detection module, a kin recognition module, and so on.
That feels pretty intuitive, actually. The mind is this survival-optimized toolkit. But hang on. If the brain is organized like that, as a collection of ancient, fixed tools, doesn't that just completely clash with what we know about neuroplasticity? We know the brain is incredibly adaptable. Regions can reorganize after an injury or with intense training. So how does a fixed, evolved module account for that kind of flexibility?
That is precisely the challenge that forced the whole theory to evolve. The neuroscience data, you're right, it shows massive plasticity, and that seriously complicates the idea of rigid, innately specified modules.
So the idea isn't totally dead.
No, not at all. The contemporary view holds on to specialization, but it rejects that rigid, fixed nature. I think the key takeaway here is that functional specialization exists, but it's more of a continuum, and it operates within these dynamic distributed networks.
Okay, so how does that actually look in practice? Maybe we can use child development as an example. Instead of a region being, born specialized, a framework like interactive specialization suggests regions only become specialized through continuous interaction with other systems and the environment.
And maybe even more counterintuitive is something called neural reuse theory. This idea posits that brain circuits that originally evolved for one purpose can be, and often are, repurposed for entirely new functions later on.
Can you give us an example of that? That sounds like the brain is being incredibly efficient, maybe a little lazy.
Oh, highly efficient. A really prominent example involves the brain circuits for spatial navigation, for literally mapping your environment. Research suggests those same circuits, which handle spatial organization, are often reused or co-opted to help us with numerical thinking.
Wait, so the part of my brain that knows how to get to the grocery store also helps me do math?
In a way, yes, the underlying structure for organizing where things are seems to get reused to organize how many there are.
That makes the concept so much clearer. So the ultimate insight is that the brain is specialized, but with reusable parts, and it's constantly integrating them into new networks depending on what you need to do. And that need for flexible mapping brings us perfectly to big idea number 2, the relentless methodological quest to link cognitive functions to their neural substrates. It's really a journey from studying bumps on the head to analyzing these complex neural networks.
And that journey has a fascinating and sometimes strange history. You really have to start with the influential, if, scientifically discredited concept of functional localization. That came from Franz Gall in the early 19th century. He's the father of phrenology.
Phrenology, right, the skull bones.
Exactly. And while his method of measuring the skull was completely unfounded, he introduced this revolutionary idea that distinct cognitive traits live in distinct brain regions.
So the core idea of localization was right, even if his execution was hilariously wrong. But the real scientific validation came later in the century with those famous lesion studies.
Right, with Broca and Wernicke.
Paul Broca found damage in the left frontal lobe, crippled speech production. And then a few years later, Carl Wernicke found a region in the posterior temporal lobe that was crucial for language comprehension. That was the first reliable link.
And then in the 20th century, you get the incredible work of Wilder Penfield. During neurosurgery, he would use direct electrical stimulation on conscious patients to map out the sensory and motor cortices with just astonishing precision. These milestones absolutely confirmed specialization is real.
But mostly for, primary sensory and motor functions. Today, we're trying to map much more complex cognition, which means we need multi-strategy approaches. We map structure with MRI, we map the wiring with DTI, and we do functional mapping with fMRI to see what's active during a task.
And yet, despite all this incredible power, modern mapping runs straight into what I think is one of the most critical conceptual stumbling blocks in the whole field,
Which is?
the reverse inference problem.
Okay, we need to spend a minute on this because it defines how we should interpret all those colorful brain scans you see in the news. You're saying reverse inference is a major logical flaw.
It is. Let me give you an analogy. Let's say you see a person sweating. That's your observation.
Okay, sweating. Got it.
And you know that running causes sweating, so you conclude that the person must be running. That's a reverse inference.
But they could also be sitting in a sauna or have a fever or being a public speech. Sweating doesn't prove they were running.
Precisely. Now apply that to the brain. We know that performing process A, let's say, feeling social pain, consistently activates region X. The reverse inference problem happens when we see activation in region X and then conclude definitively that the person must be feeling social pain.
Because that region could be activating for lots of other reasons.
Too many other reasons. Physical pain, error monitoring, general conflict. You can't draw a singular conclusion. It's a logical asymmetry. Process A activates region X, but activation of region X does not prove process A. It means we have to treat those neat, colorful blobs on fMRI scans with immense caution.
And that problem is made even worse by other challenges, right? Like individual variability and context sensitivity.
Absolutely. The sources highlight that everyone's brain is a bit different structurally and functionally, and a region's role can change depending on the network it's currently talking to. A brain's function is defined by its dynamic state, not just its static location.
That context sensitivity really shows the profound need for technology that can observe this dynamic system in action. And that's our third big idea. The whole suite of technological innovations that let us peek inside the living, working brain.
And these technologies really fall into three key buckets: structural, functional, and what we call interventional. So structurally, you have the high-definition anatomical pictures from MRI, and then the incredible detail of DTI diffusion tensor imaging, which literally maps the brain's wiring diagram by tracking water diffusion along white matter pathways.
The brain's highways. And then the functional technologies, which is where the field just exploded. You have fMRI measuring blood flow, PET scans, and then technologies that measure electrical activity like EEG and MEG.
And these functional methods led to one of the most surprising discoveries of the last 20 years: large-scale brain networks. Specifically, the discovery of the default mode network, or DMN.
Right. Before the DMN, The general assumption was that the brain was pretty quiet until you gave it a specific task to do.
Exactly. But what the DMN showed was that certain areas, like the medial prefrontal cortex, actually become more active when people are just lying still, resting, or daydreaming. They quiet down when you start an external task.
Which completely overturned that idea of the brain as just a reactive machine.
It did. It showed the brain is intrinsically organized around internal, stuff-retrieving memories, planning for the future, self-referential thought. The DMN and other networks, like the frontoparietal control network, prove that cognition emerges from the coordinated dance of these vast, distributed systems.
That is profound. But all these amazing functional methods, they still face that core limitation you mentioned earlier, that trade-off.
The trade-off between spatial resolution and temporal resolution, it is the ultimate methodological constraint.
So let's break that down again. With fMRI, you're measuring blood flow changes, which are slow, so it has poor temporal resolution. We know where the activity is happening with great precision.
Great spatial resolution.
But not exactly when it happened.
And conversely, with EEG, you're measuring electrical activity right at the scalp. Electricity is fast, so you get phenomenal millisecond temporal precision. You know exactly when the thought occurred.
Yeah.
But the signal gets blurred by the skull and tissue, so you have very limited spatial resolution. You know the timing, but you can't be sure of the precise source.
So it sounds like functional imaging really just gives us correlations. Region A and task B happen at the same time. But to prove causality, that A caused B, you need a whole different set of tools.
And that's where the interventional methods come in. This is why transcranial magnetic stimulation, TMS, is so vital. If fMRI is great for correlation, TMS is designed to test causal links. It uses a magnetic coil to induce a tiny electrical current, which can momentarily disrupt or inhibit a targeted region. It's like creating a temporary, reversible, virtual lesion.
So if I use TMS to briefly knock out region X and suddenly my subject can't perform task B, then I have pretty strong causal evidence that region X is necessary for that task.
Precisely. Unlike just watching a region light up with fMRI, TMS lets you poke the system and see what happens. It lets you test necessity. These interventional methods, combined with imaging, that's really the frontier. Multimodal approaches are the only way forward to get past those spatial or temporal trade-offs.
That brings us to the end of our deep dive into the architecture of the cognitive brain. Before we let you go, here are the essential learning outcomes for quick recall.
Okay, 4 key points to take away. First, cognitive architecture is defined by both specialization and integration. The brain uses specialized regions, but they're all interwoven into these dynamic, large-scale networks that shift depending on the context. Second, classical modularity has been radically refined. Fodor's idea of encapsulated systems was foundational, but contemporary views really emphasize plasticity and network dynamics, leading to ideas like neural Reese, where old circuits get new jobs.
Number 3.
Third, brain mapping relies on multimodal tools but faces logical pitfalls. Modern mapping with fMRI is really complicated by the reverse inference problem, which is a huge caution against equating a lit-up region with one specific mental process.
And final.
And fourth, technological choices always involve trade-offs. Researchers have to navigate that core constraint between high spatial resolution, which is knowing where, and high temporal resolution, which is knowing when. This is pushing the field toward combining techniques.
An excellent synthesis of a very complex landscape.
It seems the core challenge now is moving beyond just localization, the where, to understanding the dynamic, interactional how. It's about how these specialized parts interact predictively to produce the incredible fluidity of human thought.
And that leads perfectly to our final provocative thought for you to explore on your own. If we start thinking of the brain not as just reacting to information, but instead through the framework of predictive processing, this idea that the brain is constantly generating and testing hypotheses about what's about to happen, how does that framework challenge those traditional hard boundaries we draw between perception, cognition, and action? It ties right back to that specialization versus integration tension we talked about, and it makes you wonder how separate those rules really are in your own daily experience. Flying the mull over until our next deep dive. Thanks for joining us.